The following content is provided under a Creative Commons license. Your support will help MIT OpenCourseware continue to offer high-quality educational resources for free. To make a donation or view additional materials from hundreds of MIT courses, visit MIT OpenCourseware at ocw.mit.edu.
All right, we should get started. So it's good to be back. We'll be discussing DNA sequence motifs. We were. If you're wondering, yes, the instructors were at the awards on Sunday. It was great. The pizza was delicious. Okay. So today we're going to be talking about DNA and protein sequence motifs, which are essentially the building blocks of regulatory information, in a sense. Before we get started, I wanted to just see if there are any questions about material that Professor Gifford covered from the past couple days. No guarantees I'll be able to answer them, but just sort of general things that, related to transcriptome analysis or PCA, anything. Hopefully you all got the email that he sent out about basically what you're expected to get. So at the level of the document that's posted, that's sort of what we're expecting. So if you haven't had linear algebra, that should still be accessible. Not necessarily all the derivations. Any questions about that? Okay. So as a reminder, team projects, your aims are due soon. We'll post there's been a request for more detailed information on what we'd like in the aims. So we'll post something more detailed on the website this evening and probably extend the deadline a day or two just to give you a little bit more time on the aims. So after you submit your aims, this is students who are in the taking the project component of the course, then your team will be assigned to one of the three instructors as sort of a mentor, advisor, and we will schedule a time to meet with you in the next week or two to discuss your aims, just to sort of assess the feasibility of the project and so forth before you sort of launch into it. Okay? Alrighty. So any questions from past lectures? All right, so today we're going to talk about modeling and discovery of sequence motifs. We'll give an example of a particular algorithm that's used in motif finding called the Gibbs sampling algorithm. It's not the only algorithm. It's not even necessarily the best algorithm. It's pretty good. It works in many cases. It's an early algorithm, but it's interesting to talk about because it sort of illustrates the problem in general, and also it's an example of a stochastic algorithm, an algorithm where what it does is determined sort of at random to some extent, and yet still often converges to a particular answer. So it's interesting from that point of view. And we'll talk about a few other types of motif finding algorithms, and we'll do a little bit on statistical entropy and information content, which is a handy way of describing motifs, and talk a little bit about parameter estimation as well, which is critical when you have a motif and you want to build a model of it to then discover additional instances of that motif. So some reading for today. So I posted some Nature Biotechnology primers on motifs and motif discovery, which are pretty easy reading. The textbook, chapter 6, also has some good information on motifs. Encourage you to look at that. And I've also posted the original paper by Bailey and Elkin on the meme algorithm, which is kind of related to the Gibbs sampling algorithm, but uses expectation maximization. And so it's a really nice paper. Take a look at that, and I'll also post the original Gibbs sampler paper later today. And then on Tuesday, we're going to be talking about Markov and hidden Markov models. And so take a look at the primer on HMMs, as well as there is some information on HMMs in the text. It's it's not really a distinct section. It's kind of scattered throughout the text. So the best approach is to sort of look in the index for HMMs and read the relevant parts that you're interested in. And if you really want to understand the mechanics of HMMs and how to actually implement one in depth, then I strongly recommend this Rabiner tutorial on HMMs, which is posted. So everyone, please read that. I will use the same notation to the extent possible as the Rabiner paper when talking about some of the algorithms used in HMMs in lecture. So it should synergize well. Okay. All right. So what is a sequence motif? So in general, it's a pattern that's common to a set of DNA, RNA, or protein sequences that share a biological property. So for example, all of the binding sites of the MYC transcription factor, there's probably a pattern that they share, and that you'd call that the motif for MYC. Can you give some examples of where you might get DNA motifs or protein motifs? Anyone have another example of a type of motif that would be interesting? What about one that's defined on function? Go ahead. What's your name?
Dan.
Dan.
Phosphorylation.
Yeah. So each kinase typically has a certain sequence motif that determines which proteins it will phosphorylate. Right? Other examples? Yeah. So in that case, you might determine by functionally. You might purify that protein, incubate it with a pool of peptides, and see what gets phosphorylated. For example. Yeah, in the back.
Tages and promoter sites.
What was the first one?
Promoter sites? Oh, that was my name.
Yeah, okay. Sorry. So promoter motifs, sure. Some examples?
Like for cooperative binding, transcription binding sites and stuff.
Yeah, and so you would identify those how?
By looking at sequences upstream of genes and seeing what different sequences have in common. Right.
So I think there's at least four ways I can think of identifying those types of motifs. That's probably one of the most common types of motifs encountered in molecular biology. So one way, you take a bunch of genes where you've identified the transcription start site, you just look for patterns, short subsequences that they have in common. That might give you the Tata box, for example. Another way would be, what about comparative genomics? You take each individual one, look to see which parts of that promoter are conserved. That could also help you refine your motifs. Protein binding, you could do ChIP-seq, that could give you motifs. And what about a functional readout? You clone a bunch of random sequences upstream of a luciferase reporter, see which ones actually drive expression, for example. So that would be another. Yeah, absolutely. So there's a bunch of different ways to define them. So, and then in terms, when we talk about motifs, there are several different sort of models of increasing resolution that people use. So people often talk about the consensus sequence. So you say the Tata box, which of course is the actual, describes the actual motif, TATAAA, something like that. But that's really just the consensus of a bunch of Tata box motifs. You rarely find the perfect consensus in real promoters. The real naturally occurring ones are usually one or two mismatches away. So that doesn't fully capture it. So sometimes you'll have a regular expression. So an example would be if you were describing mammalian 5' splice sites, you might describe the motif as GT A or G A GT, or sometimes abbreviated GT R A GT, where R is shorthand for either purine nucleotide, either A or G. Those are, or you can have an N. Some motifs you could have GT NN GT or something like that. Those can be captured often by regular expressions in a language, a scripting language like Python or Perl. Another very common description of motifs would be a weight matrix. So you'll see a matrix where the width of the matrix is the number of bases in the motif, and then there are 4 rows, which are the 4 bases. We'll see that in a moment. Sometimes these are described as position-specific probability matrices or position-specific score matrices. We'll come to that in a moment. And then there are more complicated models. So it's increasingly becoming clear that the simple weight matrix is too limited and doesn't capture all the information that's present in motifs. Okay, so yeah, we talked about where the motifs come from. These are just some examples. So I think I talked about all of these except for in vitro binding, right? So in addition to doing a ClipSeq where you're looking at the binding of the endogenous protein, you could also make recombinant protein, incubate that with a random pool of DNA molecules, pull down, and see what binds to it, for example. Okay. So why are they important? So they're important sort of for obvious reasons, that they can identify proteins that have a specific biological property of interest, for example, being phosphorylated by a particular kinase, or promoters that have a particular property, that is, that they're likely to be regulated by a particular transcription factor, et cetera. And ultimately, if you're very interested in the regulation of a particular gene, knowing what motifs are upstream and how strong the evidence is for each particular transcription factor that might or might not bind there can be very useful in understanding the regulation of that gene. And they're also going to be important for efforts to model gene expression. So a goal of systems biology would be to predict, from a given starting point, if we introduce some perturbation, for example, if we knock out or knock down a particular transcription factor or overexpress it, how will the system behave? So you'd really want to be able to predict how the occupancy of that transcription factor would change. You'd want to know first where it is at endogenous levels, and then how its occupancy at every promoter will change when you perturb its levels, and then what effects that will have on the on expression of downstream genes. So these sorts of models all require really accurate descriptions of motifs. Okay, so these are some examples of protein motifs. Anyone recognize this one? What motif is that? So this has X's. X's would be degenerate positions, and C's would be cysteines, and H's histidine, of course. What is this? What is this describing? If a protein has this, what can you predict about its function?
Zinc finger.
Zinc finger, right. So this is a motif commonly seen in DNA binding transcription factors, and it coordinates the zinc. What about this one? Any guesses on what this motif is? So this is quite a short motif. Yeah.
That's phosphorylation.
Phosphorylation site, yeah. And how do you know that?
The serine is conserved, and early next to it means it's likely to see one.
Okay, so you even know like what kinase it is. Yeah, exactly. So that's sort of view. So serine, threonine, and tyrosine are the residues that get phosphorylated, and so if you see a motif with a serine in the middle, it's a good chance it's a phosphorylation site. Here are some, you can think of them as DNA sequence motifs because they occur in genes, but they, of course, function at the RNA level. These are the motifs that occur at the boundaries of mammalian introns. So this first one is the 5' splice site motif. So these would be the bases that occur at the last 3 bases of the exon. The first 2 of the intron here are almost always GT, and then you have this position that I mentioned here that's almost always A or G position, and then some positions that are biased for A, biased for G, and then slightly biased for T. And that is what you see when you look at a whole bunch of 5' ends of mammalian introns. They have this motif. So some will have better matches or worse to this particular pattern, and that's sort of the average pattern that you see. And it turns out that in this case, the recognition of that site is not by a protein per se, but it's by a ribonucleoprotein complex. So there's actually an RNA called U1 snRNA that base pairs with the 5' splice site, and its sequence, or part of its sequence, is perfectly complementary to the consensus 5' splice site. So we can understand why 5' splice sites have this motif. They're evolving to have a certain degree of complementarity to U1 in order to get efficiently recognized by the splicing machinery. Then at the 3' end of introns, you see this motif here. So here's the last base of the intron, a G, and then an A before it. Almost all introns have end with AG. Then you have a pyrimidine ahead of it. Then you have basically an irrelevant position here at -4, yeah, which is not strongly conserved. And then a stretch of residues that are usually but not always pyrimidines, called the pyrimidine tract. And in this case, the recognition is actually by proteins rather than RNA. And there are two proteins one called U2AF65 that binds the pyrimidine tract, and one U2AF35 that binds that last YAG motif. And then there's an upstream motif here that's just upstream of the 3' splice site that is quite degenerate and hard to find, called the branch point motif. OK, so let's take an example. So the 5' splice site is a nice example of a motif because you can uniquely align them, right? You can sequence cDNAs, sequence genomes, align the cDNA to the genome. That tells you exactly where the splice junctions are. And you can take the exons that have a 5' splice site and align the sequences, aligned to the exon-intron boundary and get a precise motif. And then you can tally up the frequencies of the bases and make a table like this, which we would call a position-specific probability matrix. And what you can then do to predict additional, say, 5' splice site motifs in other genes, for example, genes where you didn't have good cDNA coverage because, let's say, they're not expressed in the cells that you analyzed, you could then make this odds ratio here. Okay, so here we have a candidate sequence. So the motif is of length, it's 9 positions, often numbered like -3 to -1 would be the exonic parts of this, and then +1 to +6 would be the first 6 bases of the intron. That's just the convention that's used. I'm sure it's going to drive the computer scientists crazy because we're not starting at 0, but that's usually what's used in the literature. And so we have a 9-base motif, and then we're going to calculate the probability of generating that particular sequence S, given plus, meaning given our foreground or motif model, okay, as the product of the probability of generating the first base of the sequence S1 using the column probability in the -3 position. Okay, so if the first base is AC, for example, that would be 0.4, and then the probability of generating the second base in the sequence using the next column, and so forth. Okay, so it's basically just if you made a vector for each position that had a 1 for the base that occurred at that position and a 0 for the other bases, and then you just did the dot product of that with the matrix, you get this. Okay, so we multiply probabilities. So that is assuming independence between positions. Okay, and so that's a key assumption. Weight matrices assume that each position in the motif contributes independently to the overall strength of that motif, and that may or may not be true. They don't assume that it's homogeneous. That is, you have usually in a typical case different probabilities in different columns. So it's inhomogeneous, but assumes independence. And then you often want to use a background model. For example, if your genome composition is 25% of each of the nucleotides, you could just have a background probability that was equally likely for each of the 4, and then calculate the probability S given minus of that generating that particular 9mer under the background model, and take the ratio of those two. And the advantage of that is that then you can find sequences that are sort of, that ratio, it could be 100 times more like a 5' splice site than like background, or 1,000 times, or you have some sort of scaling on it. Whereas if you just take the raw probability, it's going to be something that's on the order of 1/4 to the 9th, so some very, very small number that's a little hard to work with, right? Okay, so when people talk about motifs, they often use language like exact or precise versus degenerate, strong versus weak, good versus lousy, depending on the context, who's listening. An example of these would be a restriction enzyme. You often say, restriction enzymes have very precise sequence specificity. They only cut, EchoR1 only cuts at GAA, TTC, whereas, TATA binding protein is somewhat more degenerate. It'll bind to a range of a range of things. So I use degenerate there. You could say it has a weaker motif. You'll often, if you want to try to make this precise, then the language of entropy and information offers additional terminology like high information content, low entropy, et cetera. Okay, so let's take a look at this as perhaps a more natural or more precise way of describing what we mean here. So imagine you have a motif. We're going to do a motif of length 1. Okay, just keep the math super simple. Okay, but you'll see it easily generalizes. So you have probabilities of the 4 nucleotides that are pk. Okay, and you have background probabilities qk, and we're going to assume that those are all it's uniform. They're all a quarter. Okay, so then the statistical or Shannon entropy of a probability distribution or vector of probabilities, if you will, is defined here. So h of q, where q is a distribution, or in this case a vector, is defined as minus the summation of qk log qk in general, and then if you want it to be in units of bits, you'd use log base 2. Okay, so how many people have seen this equation before? Like half, I'm going to go with. Okay, good. So who can tell me why, first of all, is this a positive quantity, negative quantity, non-negative, or what? Yeah, go ahead.
Log qk is always going to be negative, and so therefore you have to take the negative of the sum of all of negatives to get a positive number.
Right. So this in general is a non-negative quantity because we have this minus sign here, right? We're taking logs of things that are between 0 and 1, so they tend to be negative. The logs are negative, right? Okay, and then what would be the entropy if I say that the distribution Q is this 0, 1, 0, 0, meaning it's a motif that's 100% C. What is the entropy of that? What was your name?
William.
William.
So the entropy would be 0 because the vector is deterministic, but there's no uncertainty.
Right. And when you do the math, you'll get, for the C term, you'll have, so you have minus, you'll have a sum. You'll have 3 terms that are 0 log 0. It might crash your calculator, I guess. And then you'll have one term that's 1 log 1, right? And so 1 log 1, that's easy. That's 0, right? This, you could say, is undefined, but using L'Hopital's rule, by continuity, x log x, you take the limit as x gets small, is 0. Okay? So this is defined to be 0 in information theory. Okay? And this is always 0. Okay? So that comes out to be 0. So it's deterministic. So entropy is a measure of uncertainty, and so that sort of makes sense. If you know what the base is, there's no uncertainty. Entropy is 0. So what about this vector? A quarter, 25% of each of the bases. What is h of q? Anyone? I'm going to make you show me why. So. Anyone? Want to accept this? Levi, yeah.
I think it's 2.
2, okay. Can you explain?
Because the log of fourth is always going to be -2. And then you're multiplying that by a fourth, so you're getting half for each and adding that up equals 2.
Right. In the sum, there are going to be 4 terms that are 1/4 times log of a quarter. This is -2. Quarter times -2 is -1/2. 4 times -1/2 is -2, and then you change the sign because there's this minus in front here, right? So that equals 2, okay? And what about this one? Anyone see that one? This is a coin flip, basically, right? Either A or G, each equally likely. Anyone? Levi, you want to do this one again?
That's 1.
Okay. And why?
Because you have 2 terms of 0 log 0, which is 0, and 2 terms of 1/4, 2 terms of 1/2 times log of 1/2 would be -1. So you end up with 2 halves.
Yeah, so 2 terms like that, and then there's going to be 2 terms that are something that's turns out to be 0, right, of 0 log 0, right, and then there's a minus in front, okay? So that will be 1, okay. So a coin flip has 1 bit of information. So that's basically what we mean. If you have a fair coin and you don't know what the outcome, we're going to call that 1 bit. And so a base that could be any of the 4 equally likely has twice as much uncertainty, okay? All right, so, and this is related to the Boltzmann entropy that you may be familiar with from statistical mechanics, which is the log of the number of states, right? In that if you have n states and they're all equally likely, then it turns out that the Shannon entropy turns out to be log of the number of states. Okay, we saw that here. 4 states equally likely, it comes out to be log of 4 or 2, right? And that's true in general. All right, so you can think of this as a generalization of Boltzmann entropy if you want to. Okay, so why, let's see, why did he call it entropy? So it turns out that Shannon, who was developing this in the late 40s, as sort of developing a theory of communication, had some, scratched his head a little bit, and he talked to his friend John von Neumann, okay, none other than him, involved in, inventing computers, and he says, my concern was what to call it. I thought of calling it information, but the word was overly used. Okay, so back in 1949, information was already overused, and so I decided to call it uncertainty. And then he discussed it with John von Neumann, and he had a better idea. He said, you should call it entropy. In the first place, your uncertainty function has already been used in statistical mechanics under that name, so it already has a name. In the second place, and more important, nobody knows what entropy really is. So in a debate, you'll always have the advantage. Okay? So keep that in mind. So start, after you've taken this class, just start throwing it around, okay? And you will win a lot of debates. All right. So so how is information related to entropy? So the way we're going to define it here, which is how it's often defined, is information is reduction in uncertainty, okay? So if I'm dealing with a unknown DNA sequence, the lambda phage genome, and it has 25% of each base, if you tell me I'm going to send you 2 bases, I have no idea. They could be any pair of bases. My uncertainty is 2 bits per base, okay, or 4 bits before you tell me anything. If you then tell me it's the TA motif, which is always T followed by A, then now my uncertainty is 0, okay? So the amount of information that you just gave me is 4 bits. You reduce my uncertainty from 4 bits to 0, okay? So we define the information at a particular position as the entropy before, like before meaning the background. The background is sort of your null hypothesis, minus the entropy after. So after you've told me that this is an instance of that motif and it has a particular model, okay? So in this case, we have, you can see the entropy is going to be entropy before. This is just h of q right here, this term, and then minus this term, which is h of p, okay? So if it's uniform, we said h of q is 2 bits per position, and so then it's just, you just take, so the information content of a motif is just 2 minus the entropy of that motif model, okay? So in general, it turns out if the positions in the motif are independent, then the information content of the motif is 2w minus h of the motif, where w is the width of the motif. Okay, so for example, the entropy of the motif we said the entropy of this is 2 bits, right? Therefore, the information content is what? If this is our, let's say this is our p, okay? This is our motif. It's very degenerate. What is its information content? 0. Why is it 0? Who is that? Yeah, Matt.
Because the information content of that is 0, and then the information content of the null hypothesis, so to say, is 0. So, sorry, both of them are 2. The 2 minus 2 is 0.
The entropy, the background is 2, and the entropy of this is also 2. So 2 minus 2 is 0, right? Okay. And what about this? Let's say this was our motif. It's a motif that's either A or G. We said the entropy of this is 1 bit. So what is the information content of this motif?
1.
1. Why is it 1? Somewhere in the middle.
Background is 2. The entropy here.
Yeah. Background is 2. Entropy is 1. It's 1. Okay. And what about if I tell you it's the EchoR1 restriction enzyme? So it's GAATTC, a 6-base motif, precise. It has to be those bases. What is the information content of that motif? Yeah.
It's 12.
12. 12 what?
12 bits.
12 bits. And why is that?
Because the background is 2 times 6. So 6 bases and 2 bits for each.
Mm-hmm.
And you have all the bases are determined in the specific restriction enzyme site. So the information of that is, the entropy of that's 0. So 12 minus 0 is 12.
Right. The entropy of that motif is 0. You imagine 4,096 possible 6mers. One of them has probably 1. All the others have 0. You're going to have that big sum. They're all going to be. It's going to come out to be 0. Okay. So why is this useful at all, or is it? Okay. One of the reasons why it's useful. Sorry, that's on a later slide. Just hang with me, and it will be clear why it's useful in a few slides. Okay. But for now, we have a description of information content. So the EchoR1 site has 12 bits of information. A completely random position has 0, and a short, like a 4-cutter restriction enzyme would have 2 times 4, or 8 bits of information, right, and an 8-cutter. So you can see as the restriction enzyme gets longer, more information content. Okay. All right. So let's talk about the motif finding problem, and then we'll return to the usefulness of information content. So can everyone see the motif that's present in all these sequences? Maybe, if anyone can, please let me know. But probably can't. Okay, now, what now? These are the same sequences, but I've aligned them. Can anyone see a motif? Yeah, I heard some G's, right? So there's this motif that's it's over here. It's pretty weak and pretty degenerate. There's definitely some exceptions, but you can definitely see that a lot of the sequences have like at least GGC, possibly an A after that. Okay, right. So this is the problem that we're dealing with, right? You have a bunch of promoters, and the restriction side, the transcription factor that binds may be fairly degenerate, maybe because it likes to bind cooperatively with several of its buddies, and so it doesn't have to have a very strong instance of the motif present. And so it can be quite difficult to find. Okay, so that's why there's a real bioinformatics challenge. Motif finding is not done by lining up sequences by hand and drawing boxes, although that's how the first motif was found, the Tata box. That's why it's called the Tata box, because someone just drew a box in a sequence alignment. But these days, you need a computer to find, most motifs require some sort of algorithm to find. Okay, all right. So yeah, and like I said, it's essentially a local multiple alignment problem, right? You want a multiple alignment, but it doesn't have to be global, right? It just can be local. It can be just over a sub-region. All right. So they're basically at least three different sort of general approaches to the problem of motif finding. One approach is the so-called enumerative or dictionary approach. Okay, and so in this approach, you say, well, we're looking for a motif of length 6 because this is a leucine zipper transcription factor that we're modeling, and they usually have binding sites around 6, so we're going to guess 6, and we're going to enumerate all the 6mers. There's 4,096 DNA 6mers. We're going to count up their occurrences in a set of promoters that, for example, are turned on when you overexpress this factor, and look at those frequencies divided by the frequencies of those 6mers in some background set, either random sequences or promoters that didn't turn on, or something like that. You have two classes, and you look for statistical enrichment. Okay. This approach, this is fine. There's nothing wrong with this approach. People use it all the time. One of the downsides, though, is that you're doing a lot of statistical tests, right? You're essentially testing each 6mer. Like you're doing 4,096 statistical tests, so you have to adjust the statistical significance for the number of tests that you do, and that can reduce your power. So that's one sort of main drawback. The other reason is that maybe you don't see, maybe this protein binds a rather degenerate motif, and a precise 6mer is just too precise. None of them will occur often enough. You really have to have sort of a degenerate motif that's, C, R, Y, G, Y. That's really, the motif that it binds to. And so you don't see it unless you use something more degenerate. So you can use, you can generalize this to use regular expressions, et cetera. And it's a reasonable approach. Another approach is, that we'll talk about in a moment, is probabilistic optimization, where you sort of wander around the possible space of possible motifs until you find one that looks strong, okay? And we'll talk about that. And then there are sort of deterministic versions of this, like mean, okay? So, which, yeah, we'll get to. We're going to focus today on this second one, mostly because it's sort of a little bit more mysterious and interesting as an algorithm. Okay. And it's also NAT use. So, if the motif landscape looked like this, where imagine sort of all possible motifs, you've somehow come up with a 2D lattice of the possible motif sequences, and then the strength of that motif, or the degree to which that motif description corresponds to the true motif, is represented by the height here. Then there's basically this one optimal motif, and the closer you get to that, the better fit it is. Then our problem is going to be relatively easy. But it's also possible that it looks something like this. There's a lot of sort of decoy motifs or weaker motifs that are only slightly enriched in the sequence space, and so you can easily sort of get tripped up if you're wandering around randomly. So that's sort of we don't know a priori, and it's probably not as simple as the first example. And so that's one of the sort of issues that motivates these stochastic algorithms. So just to sort of put this in context, so the Gibbs motif sampler that we're going to be talking about is a Monte Carlo algorithm. So that just means it's an algorithm that basically does some random sampling somewhere in it, so that the outcome that you get isn't necessarily deterministic. Often, you run it different times and you actually get different outputs, which can be a little bit disconcerting and annoying at times, but turns out to be useful in some cases. Okay? There's also sort of a special case of a Las Vegas algorithm where it knows when it got the optimal answer, but in general, not. Okay? In general, you don't know for sure. So the Gibbs motif sampler is basically a model where you have a likelihood for generating a set of sequences S. Okay? So imagine you have 40 sequences that are bacterial promoters, each of length of 40 bases long, let's say. Okay? That's your S. Okay? And so what you want to do then is consider a model that there is a particular instance of a motif you're trying to discover at a particular position in each one of those sequences, not necessarily the same position, just some position in each sequence. And we're going to describe the composition of that motif by a weight matrix. Okay, one of these matrices that's of width W and then has the 4 rows specifying the frequencies of the 4 nucleotides at that position. Okay, and so the setup here is that you want to calculate or think about the probability of S comma A. S is the actual sequences, And A is basically a vector that specifies the location of the motif instance in each of those 40 sequences. Okay? And that is given by, you want to calculate that conditional on capital theta, okay, which is our weight matrix. So that's going to be, in this case, I think I made a motif of length 8, that's shown there in red. There's going to be a weight matrix of length 8, and then there's going to be some sort of background frequency vector that might be the background composition in the genome, of E. coli DNA, for example. Okay, and so then the probability of generating those sequences together with that particular locations is going to be proportional to this. Basically, use the theta, the little theta background vector for all the positions except the specific positions that are inside the motif, starting at position, it's sort of AK here. Okay, and then you use the particular column of the weight matrix for those 8 positions, and then you go back to using the background probabilities. Question? Yeah.
Is this for finding motifs based on other known motifs, or is this de novo?
No, we're doing, I'm sorry, I should have clarified that. We're doing de novo motif finding. We're going to tell the algorithm, we're going to give the algorithm some sequences of a given length, or it can even be variable lengths, and we're going to give it a guess of what the length of the motif is. So we're going to say 8. We think it's 8. That could come from structural reasons, or often you really have no idea. So you just guess that, a lot of times it's kind of short, so we're going to go with 6 or 8, or you try different lengths. Okay, totally de novo motif finding. Okay, so how does this algorithm work? So you have n sequences of length L. You guess that the motif has width W. You choose starting positions at random. Okay, so this A vector of the starting position in each sequence, we're going to choose completely random positions within the end sequences. They have to be at least W before the end, right? So we'll have a whole motif. That's just sort of an accounting thing to make it simpler. And then you choose one of those sequences at random, okay? Say the first sequence. You make a weight matrix model of width W from the instances in the other sequences. Okay, so for example, actually I have slides on this, so we'll just do it with the slides. You'll see what this looks like in a moment. And so you have instances here in this sequence, here in this one, here. You take all those, line them up, make a weight matrix out of those, and then you score the positions in sequence 1 for how well they match. Okay. So let me just do this more. So you have these are your motif instances. Again, totally random at the beginning. Then you take, you build a weight matrix, okay, from those by lining them up and just counting frequencies. Then you pick a sequence at random. Yeah, your weight matrix doesn't include that sequence typically. And then you take your theta matrix, okay, and you slide it along the sequence. Okay, you consider every subsequence of length W, the one that goes from 1 to W, the one that goes from 2 to W plus 1, et cetera, all the way along the sequence until you get to the end, and you calculate the probability of that sequence using that likelihood that I gave you before. Okay, so it's basically the probability of generating the sequence where you use the background vector for all the positions except for the particular motif instance that you're considering, and use the motif model for that. So does that make sense? Okay, so if you happen to have a good looking occurrence of the motif at this position here in the sequence, then you would get a higher likelihood. Okay. So for example, if the motif was let's say it's 3 long, and it happened to favor it happens to favor A, C, G, then if you have a sequence here that has AA, let's say it's got TTT, okay, that's going to have a low probability in this motif. It's going to be 0.1 cubed, right? And then if you have an occurrence of, say, ACT, that's going to have a higher occurrence. Okay, it's going to be 0.7 times 0.7 times 0.1, so quite a bit higher, right? So you start, it'll be low for this triplet here, so we'll put a low value here. TTA is also going to be low, right? TAC, also low, but ACT, that matches 2 out of 3 to the motif. You know, it's going to be a lot better, and then CT, it's going to be low again, et cetera. So you just slide this along and calculate probabilities. And then what you do is you sample from this distribution. Okay, so these probabilities don't necessarily sum to 1, but you renormalize them so that they do sum to 1. You just add them up, divide by the sum. Now they sum to 1, and now you sample those sites in that sequence according to that probability distribution. Okay. So yeah, so like I said, in this case, you might end up sampling, that's the highest probability site. So you might sample that, but you also might sample, you know, one of these other ones. It's unlikely you would sample this one because that's, you know, very low, but you actually sometimes sample one that's not so great. Okay. All right, so you sample the starting position in that sequence, and you basically, wherever you had originally assigned in sequence 1, now you move it to that new location. Okay. We've just changed the assignment of where we think the motif might be in that sequence. Okay? And then you choose another sequence at random from your list. Often you go through the sequences sequentially, and then you make a new weight matrix model. Okay? So how will that weight matrix model differ from the last one? Well, it'll differ because the instance of the motif in sequence 1 is now at a new location in general. I mean, you might have sampled the exact same location you started, but in general, it'll move, and so now you'll get a slightly different weight matrix. Most of the data going into it, n-1, is going to be the same, but one of them is going to be different, so it'll change a little bit. Okay, and then you make a new weight matrix, and then you pick a new sequence, you slide that weight matrix along that sequence, you get this distribution, you sample from that distribution, and you keep going. Okay. Yeah, this was described by Lawrence in 1993, and I'll post that paper. Okay, so you sample proportional to that, and you update the location. So now we sampled that really high probability one, so we moved the motif over to that new orange location there. Okay. I don't know if these animations are helping at all, but okay, and then you update your weight matrix. Okay. All right. And then you iterate until convergence. So you typically have you have a set of n sequences. You go through them once. You have a weight matrix, and then you go through them again. You go through a few times. And maybe at a certain point, you end up resampling the same sites as you did in the last iteration. Okay, same exact sites. You've converged. Okay, or you keep track of the theta matrices that you get after going through the whole set of sequences, and from one iteration to the next, the theta matrix hasn't really changed much. Okay, you've converged. All right, so let's do an example of this. Here I made up a motif, and this is a sort of representation where the 4 bases have these colors assigned to them, and you can see that this motif is quite strong, right? It's really strongly prefers A at this position here, and et cetera. And I put it at the same position in all the sequences just to make life simple, okay? And then a former student in the lab, Mexdi, he implemented the Gibbs sampler in MATLAB, actually, and made a little video of what's going on. Okay, so the upper part shows the current weight matrix. Okay, so notice it's pretty random looking at the beginning, and the right parts show where the motif is, okay, or the position that we're currently considering. So this shows the position that was last sampled in the last round, okay, and this shows the probability density along each sequence of what's the probability that the motif occurs at each particular place in the sequence. Okay, and that's what happens over time. Okay, so it's obviously very fast, so I'll run it again and maybe pause it part way. Okay, so we're starting from a very random looking motif. Oops, sorry. Okay, this is what you get after not too many iterations, probably like 100 or so. Okay, now you can see your motif, your weight matrix is now quite biased, okay? It now favors this position, and so forth. And the locations of your motif, most of them are at around this position, you know, around 6 or 7 in the sequence, that's where we put the motif in, okay? But not all, some of them. And then you can see the probabilities, white is high, black is low, are, you know, in some sequences, it's very, very confident that the motif is exactly at that position, like this first sequence here. In others, you know, it's got some uncertainty about where the motif might be, okay? And then we let it run a little bit more, and it eventually converges to being very confident that the motif has this sequence, ACGT, AGCA, and that it occurs, you know, at that particular position in the sequence. Okay. So who can tell me why this actually works? Right, we're choosing positions at random, updating a weight matrix. Why does that actually help you find the real motif that's in these sequences? Any ideas? Or who can make an argument that it shouldn't work? Yeah, okay. What was your name again? Dan, yeah, go ahead.
So, I mean, it couldn't, in certain situations, you can have different like sub-motifs that are also sort of enriched, and because you're sampling randomly, you might be stuck inside those sort of like sub-values when you're searching your composition.
Yeah, that's true. So Dan's point is that you could get stuck in like suboptimal, you know, smaller or weaker motifs. So that's certainly true. So you're saying maybe this example is artificial because I had started with totally random sequences and I put a pretty strong motif, you know, in a particular place. So there were no. It's more like that mountain, that structure, right, where there's just one motif to find. So it's perhaps an easy case. But still, what I want to know is, how did it actually find that motif? He implemented exactly that algorithm that I described. Why does it tend to go toward that motif? After a long time. Remember, it's a long time. It's hundreds of iterations.
So, I mean, you're covering a lot of the sequence just through random searching of the sequence.
There are many iterations. Yeah. You're considering many possible locations within the sequences. That's true. But why does it eventually why does it converge to something?
I guess because like you're seeing your like motif more frequently than you're seeing other random motifs. So it'll hit it more frequently randomly, and therefore enrich for it, I guess.
Yeah, that's true. Can someone give a more, like, maybe more intuition behind this? Yeah.
I just had a question. Is each iteration an independent test? For example, if you iterate over the same sequence space 100 times, and you're updating your weight matrix each time, does that mean, is the updating the weight matrix also taking into account that that you're like, this is the same sample space? Is that question?
Yeah, the weight matrix, after you go through one iteration of all the sequences, you have a weight matrix, okay? You carry that over. You don't start from scratch. You bring that weight matrix back up and use that, you know, to score, let's say, that first sequence. So it keeps, the weight matrix just keeps, you know, moving around. It moves a little bit, you know, with every time you sample a sequence.
So it constantly gets stronger then.
Well, does it?
Well, I guess
Would it constantly get stronger? What's to make it get stronger or weaker? I mean, this is sort of you're sort of on the track.
It's random.
Yeah.
Then there's some probability that you're going to find this motif again, at which point it will get stronger. But if it's so I guess given enough iterations, it gets stronger if the sampling, as long as you hit like different spots at random.
Yeah. That's more. Yeah, I think there was a comment. Diego. Yeah.
Well, I mean, you can think about it as like a random walk through the landscape. Eventually, it has a high probability of hitting that motif and updating the weight matrix in the right direction, just from the probability of the motif being
Okay.
And given enough iterations, it'll convert to
Okay, let's say I had 100 sequences of length, I don't know, 30, and the width of our motif is 6, okay? So here's our sequences. We choose random positions for the start position. And let's say it was this example where the real motif, I put it like right here in all the sequences. That's where it starts, okay? So does this help? So it's 30 and 6, so there's 25 possible start positions. I just did that to make it a little easier, okay? So what would happen, like in that first iteration, what would that matrix, what can you say about what the weight matrix would look like? It's going to be a width W, columns 1, 2, 3, up to 6. We're going to give it 100 positions at random. The motif is here. Let's say it's a very strong motif. That's a 12-bit motif. So it's 100%. It's echo R1. Okay, it's that. What would that weight matrix look like after the first, in this first iteration, when you first just sample the sites at random? What kind of probabilities would it have? Equal? Okay. Perfectly equal? No.
Roughly.
Okay. Any bias? Are we likely to hit the actual motif ever in that first iteration?
No, because you have a uniform probability of sampling. Uniform at each one of the 25 positions.
Right.
Right now, you're not sampling proportional to the
That's right.
likelihood.
So the chance of hitting the motif in any given sequence is what?
1 over 25.
1 over 25. We have 100 sequences.
4.
So on average, you'll hit the motif 4 times. Right. The other 96 positions will be essentially random, right? So you initially said this is going to be uniform, right? Like on average, 25% of each base, plus or minus a little bit of sampling error, right? It could be 23, 24, 26, right? But now you point out that there's going to be 4. You're going to hit the motif 4 times on average, right? So can you say a few more?
You maybe have a slightly biased towards G on the first position.
Okay.
Slightly biased towards A on the second and third. Slightly biased towards T on the fourth and fifth, and slightly biased towards C on the sixth. So it'd be slightly higher than 25.
Yeah. So remind me your name?
I'm Eric.
Eric, okay. So Eric says that because 4 of the sequences will have a G at the first position, because those are the ones where you sample the motif, and the other 96 will have, each of the 4 bases equally likely. On average, you'll have like 24% plus 4 for G, right? Something like 28%. This will be 28% plus or minus, a little bit. And these other ones will be whatever, whatever that works out to be, like 20, what is that? Like 23 or something like that. 23-ish, right? On average. Again, it may not come out exactly like G may not be number 1, but it's more often going to be number 1 than any other base, right? And on average, it'll be more like 28% rather than 25%, right? And similarly, for position 2, A will be 28%, right? And 3, and et cetera. And then the 6 will be, C will have a little bit of a bias. Okay, so even in that first round, when you're sampling that first sequence, the matrix is going to be slightly biased toward the motif, depending how the sampling went. You might not have hit any instances of motif, right? But often it'll be a little bit, right? Okay? Is that enough of a bias to give you a good chance of selecting the motif in that first sequence?
You mean in the first iteration?
And let's just say the first random sequence I sample. No, you're shaking your head. Not enough of a bias because it's 0.28 over 0.25, right? It's a times to the sixth power, right? So it's like,
The likelihood is still close to 1.
yeah.
Like that starting odds ratio.
So it's something like 1.1 to the sixth, or something like that. So it might be close to 2, right? It might be twice as likely, but still there's 25 positions. So, you know, does that make sense? So it's quite likely that you won't sample the motif in that first round. You'll sample something else, which will take it away, right, in some random direction, right? So who can tell me like how this actually ends up working? Why does it actually converge eventually if you give it long enough? Any ideas? So the information content, what will happen to that? So the information content, if it was completely random, we said that would be uniform. That would be 0 information content, right? This matrix, which has like around 28% at 6 different positions, will have an information content that's low but non-zero, right? It might end up being like 1 bit or something, right? And if you then sample motifs that are not the motif, they will tend to reduce the information content. They'll tend to bring it back toward random, right? If you sample motifs that have locations that have the motif, what will that do to the information content? Boost it, right? So what would you expect if we were to plot the information content over time? What would that look like?
Trend upwards, but it could fluctuate
Yeah.
over the number of iterations.
Oops. Sorry. I think I blocked it here. Let me see if I can. Yeah, let's try this. I think I plotted it. Huh. Okay, never mind. Anyway, I think I wanted to keep it very mysterious, so you guys have to figure it out. So, yeah, the answer is right. That it will. Basically, what happens is you start with a weight matrix like this. A lot of times, because the bias for the motif is quite weak, a lot of times you'll sample, even for a sequence, what matters is, like if you had a sequence where the location initially was not the motif, and then you sample another location that's not the motif, that's not really going to change it. I mean, it'll change things a little bit, but not in any particular direction. What really matters is when you get to a sequence where you already had the motif, if you now sample one that's not the motif, your information content will get weaker, right? It'll become more uniform, okay? But if you have a sequence where it wasn't the motif, but now you happen to sample the motif, then it'll get stronger, right? And when it gets stronger, it'll then be more likely to pick the motif in the next sequence, right? And so on, right? So basically what happens to the information content is over many iterations, it starts near 0, and it can occasionally go up a little bit. And then once it exceeds a threshold, it goes like that, okay? So what happens is it stumbles onto a few instances of the motif that bias the weight matrix. And if they don't bias it enough, it'll just sort of fall off that. It's like trying to climb the mountain, but it's walking kind of in a random direction. So sometimes it'll turn around and go back down. But then when it gets high enough, it'll sort of be obvious. Then you have, like once you have say a 20 times greater likelihood of picking that motif than any other sequence, most of the time you will pick it, and very soon it'll get strong. It'll be stronger. In the next round, when it's stronger, you'll have a greater bias for picking the motif, and so forth. Question? Yeah.
For this specific example, n is like much greater than L minus W. How true is that for like practical examples?
Yeah, that's a very good question. So, it varies. Sometimes it depends on how commonly your motif occurs in the genome and how good your data is really, what the source of your data is. So sometimes it can be very limited. Sometimes if you do ChIP-seq, you might have 10,000 10,000 peaks that you're analyzing or something. So you could have a huge number. But on the other hand, if you did some functional assay that's quite laborious for a motif that drives luciferase or something, and you can only test a few, you might only have 10. So it's really it varies all over the map. So that's a good question. We'll come back to that in a little bit. Simona?
Short motif, does it make sense then to reduce the number of sequences you have? Because maybe it won't converge.
Reduce the number of sequences?
Yeah.
What do people think about that? Is that a good idea or a bad idea? It's true that it might converge faster with a smaller number of sequences, but you also might not find it at all. So generally, you're losing information. So you want to have more sequences up to a certain point. So let's just do a couple more examples, and I'll come back. Those are both good questions. Okay, so here's this weak motif. So this was the one where you guys couldn't see it when I just put the sequences up. You can only see it when it's aligned, right? It's this thing with GGC here. Okay, and here's the Gibbs sampler, and what happened? Who can summarize what happened here? Yeah, David. Yeah, it didn't quite converge, right? You can see it. The motif is usually on sort of on the right side, and it found something that's like the motif, right? But it's not quite right? It's got that A. It's GAGC. It should be like GGC, so it sampled some other things, and it got sort of off track a little bit because probably by chance there were some things that looked a little bit like the motif, and it sort of was finding some instances of that and some instances of the real motif, and yeah, it didn't quite converge. And you can see this probability vectors here. They have multiple white dots in many of the rows, right? So it doesn't know. It's uncertain, so it keeps bouncing around, right? So it didn't really converge. It was too weak. It was sort of too challenging for the algorithm. Okay, so this is just a sort of a summary of the Gibbs sampler, how it works. It's not guaranteed to converge to the same motif every time. So what you generally will want to do is run it several times. And if 9 out of 10 times you get the same motif, you should trust that more. Go ahead.
Over here, are we optimizing for convergence of the value of the information content?
No. The information content is just describing. It's just a handy, single number description of how biased the weight matrix is. So it's not actually directly being optimized, right? But it turns out that this way of sampling tends to increase information content because it's sort of a self-reinforcing kind of thing. Okay. Yeah, but it's not directly doing that. However, mean more or less directly does that. So the problem with that is that where do you start, right? See, imagine an algorithm like this, but where you deterministically, instead of sampling from the positions in the sequence where it might have a motif in proportion to probabilities, you just chose the one that had the highest probability. That's more or less what meme does, okay? And so what are the pros and cons of that approach versus this one? Any ideas on this? Okay, one of the big disadvantages is that the initial choice of, how you're initially seeding your matrix matters a lot, right? That slight bias, like it might be that you had a slight bias, it didn't come out being G was number one, it was actually T was number one, just because of the, the quirks of the sampling. So what would this be like, 31 or something like that? No, I don't know. Anyway, something, it's higher than these other guys. And so then you're always picking the highest. It'll become a self-fulfilling prophecy, okay? So that's the problem with meme. And so the way that meme gets around that is it uses multiple different seeding, multiple different starting points, and goes to the end with all of them. And then it evaluates how good a model did we get at the end. And whichever was the best one, it takes that. Okay, so it actually takes longer. But then you only need to run it once because it's deterministic. You use a deterministic set of starting points. you run a deterministic algorithm and then you evaluate. Okay. Yeah, the Gibbs, it can go off on a tangent, but then because it's sampling so randomly, it often will fall off that and come back to something that's more uniform. And when it's a uniform matrix, it's really sampling completely randomly, exploring the space in an unbiased way. Tim. Yeah.
For genomes that have inherent biases that you know going in, do you like pre-calculate? Do you just like pre-calculate the weight matrix to kind of before to affect those biases? For example, if you had like 80% AT content, then you're not looking for, you know immediately that you're going to hit an ART after the first iteration. So how do you
deal with that? Yeah. Good question. Okay. All right, so let me, okay. So these are some features that affect motif finding. Okay, and actually several, I think that we've now hit like at least a few of these. So number of sequences, length of sequences, information content, and motif, and basically whether the background is biased or not. So in general, higher information content motifs or lower information content are easier to find. Who thinks higher? Who thinks lower? Okay. Someone, can you explain yourself? What's your name?
I don't know.
No? Just a guess? Okay. It was in back. Can you explain? Lower?
Low information content means that it's basically very similar. So
low information means nearly uniform. Right. Those are very hard to find. That's like that GGC one. The high information content motifs, those are the very strong ones, like that first one we looked. Those are much easier to find because when you stumble onto them, it biases the matrix more and you rapidly converge to that. Okay, so high information is easier to find. So if I have one motif per sequence, what about the length of the sequence? Is longer or shorter better? Is longer better? Who thinks shorter is better? Shorter. Can you explain why shorter?
Trying to be a smaller search space with fewer problems.
Exactly. Shorter the search space, and your motif, there's less place for it to hide. You're more likely to sample it, right? So yeah, shorter is better. If you think about like, if you have a motif like Tata, which is typically 30 bases from the TSS, if you happen to know that and you give it like +1 to -50, you're giving me a small region, you can easily find the Tata box. If you give it +1 to minus 2,000 or something, right? You may not find it. It's diluted, essentially. Okay. Number of sequences, the more the better. This is a little more subtle, as Simona was saying. It affects convergence time and so forth. But in general, the more the better. And if you guess the wrong length of your matrix, that makes it worse than if you guess the right length in either direction. Okay, for example, it's a 6-base motif, you guess 3. Okay? The information content, even if it's a 12-bit motif, there's only 6 bits that you could hope to find, right? Because you can only find 3 of those positions, right? So clearly, effectively, it's a smaller information content, much harder to find, okay? And vice versa. Yeah. Another thing that occurs in practice is what's called shifted motifs. Okay, your motif is GAATTC. Imagine in your first iteration, you happen to hit several of these sequences starting here. Like you hit the motif but off by 2, okay, at several different places. That'll bias the first position to be A, right, and the second position to be T, and so forth. And then you'll tend to find other shifted versions of that motif. You may well converge to this, ATCCNN or something like that, right? Which is not quite right. It's close, right? You're very close, but not quite right. And it's not as information rich as the real motif, right? Because it's got those 2 N's at the end instead of GA. So one thing that's done in practice is a lot of times, like every so often, the algorithm will say, what would happen if we shifted all of our positions over to the left by 1 or 2, or to the right by 1 or 2? Would the information content go up? If so, let's do that. So that's sort of another. So basically, shifted versions of the motif become effectively, they're like local, near optimal solutions. So you have to avoid them. Okay. And biased background composition is very difficult to deal with. So I will just give you one or two more examples of that in a moment and continue. So in practice, I would say the Gibbs sampler is sometimes used, or a lineace, which is sort of a version of Gibbs sampler, but probably more often people use an algorithm called MEME, which is this EM algorithm, which, like I said, is deterministic, so you always get the same answer, which makes you feel good. May or may not always be right, but you can try it out here at this website. And actually, the Franco Lab has a very nice website called Web Motifs that runs several different motif finders, including, like I said, a MEME and a lineace, which is similar to Gibbs, as well as some others. And it kind of integrates the output. So that's often a handy thing to use. You can read about them there. And then I just wanted to say a couple words. This is related to Tim's comment about the biased background. Okay, how do you actually deal with that? Okay, and this is kind of related to this notion of a mean bit score of a motif. Okay, so if I were to give you a motif model P and a background model Q, then the natural scoring system, if you wanted additive scores instead of multiplicative, you would just take the log, okay? So log P over Q, I would argue, is natural additive scores, and that's often what you'll see in a weight matrix. You'll see log probabilities or logs of ratios of probabilities, and so then you just add them up, and it makes life a bit simpler. And so then if you were to calculate what's the mean bit score, if I had a bunch of instances of motif, it will be given by this formula that's here in the upper right. So that's your score. And this is the mean where you're sampling over the probability in using the motif model probabilities. Okay? Okay, so it turns out then that if QK, your background, is uniform, motif with W, so its probability of any W mer is 1 over 4 to the W, then it's true that the mean bit score is 2W minus the entropy of the motif, which is the same as the information content of the motif using our previous definition. Okay, so that's just some sort of a handy relationship. And you can do a little algebra to show that if you want. So basically, summation pk log pk over qk, this log, you turn that into a difference, right? So that's summation pk log pk minus pk log qk, okay? And then you can do some rearrangement and sum them up, and you'll get this formula, okay? I'll leave that as an exercise. If any questions on it, you can do it next time. Okay. So what I wanted to get to is sort of this, this big question that I posed earlier. What's the use of knowing the information content of a motif? And the answer is one use is that it's true, sort of in general, that a motif with m bits of information will occur about once every 2 to the m basis of random sequence. Okay? So we said a 6-cutter restriction enzyme, EcoR1, has a information content of 12 bits. So by this rule, it should occur about once every 2 to the 12th bases of sequence. And if you know your powers of 2, which you should all commit to memory, that's about 4,000. Okay, that's 2 to the 12th is 4 to the 6th is 4,096. So it'll occur about once every 4 kb, which if you've ever cut E. coli DNA, is about right. Your fragments come out to be about 4 kb. So this turns out to be strictly true for any motif that you can represent by a regular expression, like a precise motif, or something where you just have like a degenerate, like an R or Y or N in it. It's still true. And if you have a sort of more general motif that's described by a weight matrix, then it's, you have to define a threshold, and it's sort of roughly true, but not exactly. Okay. All right, so what do you do when the background composition is biased, like Tim was saying? What if it's 80% A plus T? So then it turns out that this mean bit score is a good way to go, okay? So like I said, the mean bit score equals the information content in this special case where the background is uniform. But if the background is not uniform, then you can still calculate this mean bit score, and it'll still be meaningful, but now it's called something else. It's called relative entropy. Actually, it has several names: relative entropy, Kullback-Leibler distance is another, and information for discrimination, depending whether you're reading like the double E literature or statistics or whatever. And so it turns out that if you have a very biased composition, so here's one that's 75% AT, okay, probability of A and T are 3/8, C and G are 1/8. If your motif is just, it's a C 100% of the time, your information content by the original formula that I gave you would be 2 bits, right? However, the relative entropy will be 3 bits. Okay, if you just plug in these numbers into this formula, it'll turn out to be 3 bits. And my question is, which one better describes the frequency of C in the background sequence? Frequency of this motif. The motif is just a C. Okay? You can see that the relative entropy says that actually that's stronger than it appears. Because it's a C and that's a rare nucleotide, it's actually stronger than it appears. And so 2 to the third is a better estimate of its frequency than 2 squared. Okay, so relative entropy. So what you can do when you run a motif finder in a sequence of biased composition, you can say, like, what's the relative entropy of this motif at the end, and look at the ones that are strong. Okay, we'll come back to this a little more next time. So next time we'll talk about hidden Markov models, and please take a look at the readings, and please, those who are running projects, look for more detailed instructions to be posted tonight.